Okay, so welcome everyone. Welcome to the session of an intro to in silico drug designing. So it's a very unique topic that we'll be discussing today, and it's a very interesting topic also for many of the people who are interested in going for bioinformatics analysis. So in today's topic, what is our aim? See, our main aim is to understand or just to have a very basic notion about the concept of modeling, whether we are talking about the protein part, how the protein is folding, or what are the software tools that are present in folding a protein, or when we are talking about the in silico designing in terms of drug development, a complete drug development in the complete umbrella of drug development, we are talking about the in silico part only. And what is in silico part? We should be able to know what are the platforms that are available for performing this drug development, how actually that drug development is performed, and what is a basic concept. What is the basic concept that I will be looking for drug development? So, see, although the complete in silico drug development module is of, if I say it's of 15 to 15 days to 3 months of module, that will be absolutely true. A crash course kind of thing will be completed in 15 days, and in-depth understanding of in silico drug development is taken up as a module for 3 months or more. So what is our job today? Our job today is to summarize everything and give you at a single plate a glimpses of the things that happens in computational approaches, how we go about designing small molecules that can be taken up as a drug under clinical settings, of course after validating in the wet lab scenario, or when we are talking about the biologics, what are biologics, what is antibody therapeutics, and how these are designed based on the in silico modulation, how I can design or predict an epitope region when I'm talking about the proteinaceous part. So that should be the outcome of this study. So before directly diving into the topic, let us discuss why actually this is relevant. See, in my opinion, when I say why this is relevant, we are talking about in terms of business and in terms of ethics, right? Ethics scenario. So when I'm talking about in terms of business scenario, the complete expenditure that is invested in terms of screening the compound is minimized. Why? Because out of the complete pool of 1,000, you are choosing 10, and you are choosing them with a rationale behind your picking or choice. There is a certain logic, there is certain theoretical concepts or the values that you have determined in order to pick the 10 out of 1,000, and that leads to your selection, then refinement, and then preclinical phase studies. You cannot go directly to, these days you cannot go directly in screening 1,000 compounds. You first have to screen those compounds which are relevant for you, and that screening part or designing part or exactly mimicking that particular part is called as in silico designing. And then there are ethical drivers. See, ethical drivers, whenever you are testing any drug, suppose you want to test the 100 drugs, then those drugs, when you are testing in preclinical phases, then at certain point the in vivo testing starts. So in vivo testing, we have a basic ethics, and that basic ethics is of single line, that is refine, reduce, and replace. Refine, reduce, and replace. Whenever you are using animals in research, these are the principles that we follow. The animals should not get the painful treatment. We should not unnecessarily be using the animals for our screening purpose. So if you want to reduce the number of animals that you want to use, of course you have to choose the compounds that are most potent. And for if you are not performing in silico drug designing, then you will be choosing them by injecting the similar compounds inside the animals. So if you are injecting 100 compounds and picking one compound, then you are using multiple animals. But once you have done the in silico drug design, in silico drug development, then from that 1,000 or the hundreds, you have identified most potent 5 or 6, and that 5 or 6 you are taking the animal studies. So you are reducing the number by also refining your research, and then there is no ethical grievances that you will be having, right? So that is the thing, that when we are talking about the relevance of the animals data for the human use, it should be taken care of ethically. It should be taken care of ethically, and also the safety part the molecules that we are using that has to be clearly cut defined before going to the in vivo studies. So when we are talking about the extrapolation of the data across the species, then we say that choice of species, what we are choosing is of most important. That uh, if I say the attrition of the compounds, then attrition of compounds because of no efficiency is 30%, but because of the toxicity is almost 10%. 10% of your compound is removed because of your toxicity. So that is the reason when we say that even a compound which has shown no toxicity in animal physiological condition, and you are saying based on in vivo studies that this particular will work exactly the same in human physiologic model, then this extrapolation barrier, most of 10% of the compounds fail or are directed because of the toxicity in this scenario, because of the toxicity in this scenario. And we are talking about drugs, but when we are talking about the biologics, that prediction is next to impossible. It becomes more difficult. It becomes more difficult. So let us focus on, first of all, focus on the small molecular drugs. Then when we are talking about the small molecular drugs, what I'm talking about, I'm talking about the safety and the efficacy part. When I'm talking about the safety part, we are mostly focusing on the bystander effects. I am using a small molecule targeted to a certain cell. Now that small molecule should be targeted to that cell only if it is having off-target effects, if it is having certain metabolic toxic products, or it is clearance from the blood is very high, then there is no point of taking the drug ahead. And when we are talking about the efficacy, the efficiency, then I am focusing on the bioavailability of the drug, or I am talking about the pharmacokinetic study. So all these studies need to be done. But first of all, what we need? First of all, what we need? We are talking about things way ahead. First of all, you need a compound. You need a drug which you can call it a small molecular entity, right? Now, from where you will get the drug? There are chemical repositories, chemical databases that are fully rich source of compounds or molecules. Now, these database includes safety information of every compound. Every compound you can choose, you can go to a database and you'll get the complete toxicity profile, the complete safety information, the complete safety information. Then there are databases. These databases are, suppose I am a vendor, I'm running a company, I have designed a certain molecule, then it is my responsibility that I'll register that molecule, I'll incorporate it in chemical database, and then I will provide the complete structure also. And that you can see in the Zinc database, the complete structure is also always present. Then there are databases which provide the biological part of those chemicals also, whether these chemicals have certain activity, whether this chemical can act as certain inhibitor, or there are certain studies that been done, whether this information is retrieved from PubChem, or whether we are talking about Chem Lab, or the Kyoto database that we are talking about, the Kyoto encyclopedia that we are talking about. So all these repositories are there for us to use. They are reducing our efforts, and that's why it's very important to understand that when we are talking about chemical EMBL database, the European Molecular Biology Laboratory database, which deals with the chemical compounds, it contains the complete medicinal chemistry bio database. It contains the literature, it contains the data sets, it contains the biological databases also. So wide variety of sources are there and integrated to give you the complete information in the single page. So what we are talking about, this particular database in nutshell contains almost around 16 lakh plus compound records, which are targeting more than 10,000 targets. And their activities and publications are also listed. So you have the complete access to all the compounds, and this is how it looks. The ChemBell, I call it as ChemBell, it's a chemical EMBL database. So what it's there, you can go for ligand research. If you want to search any kind of compound, you can type the name of the compound and you can go for ligand research, or you can use the target research to incorporate the target where the target is there. You can also browse and add your chemical compounds and perform the target based studies here. there will be no issue. You can perform the smile search and you can perform the ID search from these portals also. You can search in this panel any document whether you want to go for, and then the consortiums that are being run, the databases that is there maintained by this at the platform of Campbell, it's also there. They have this Unichem, which will be unique chemical identification number is there. So there are multiple platforms. All you need to go do is go and explore. So once I've incorporated the compound, I can work with the compound. I can incorporate different ligands to it. I can go for bromination, chlorination, fluorination, addition of hydroxyl group on their surface, and can design my compound here, or can modify my compound here, and then I can see how it targets my target molecule. So the refinement part can also be done here. You can see here there is a hexane ring, there is a pentane ring which I want to incorporate. So it's just, and best thing is if you are not aware how to use it, they will provide you the training module also. They will provide you the training module also. So wherever you find the difficulty, it is very easy to resolve. It's very easy to resolve. And more you practice, more you attain. More you practice, more you attain. So what can I do with this particular model? What can I do with this particular model? See, what can I do? There are two major things that I can compare the structural similarity between the two compounds. That is of most importance. If there is a compound that is there and showing toxicity, I can find its structural analog, which is of low toxicity and higher bioavailability. So what I can do, I can map the structural characteristics to the properties, whether that change in the structure will lead to change in the property, that I can do. I can also do whether this compound is having any molecular interactions. Maybe if the compound is interacting with certain amino acids, or it is interacting with the hydroxyl group on the polysaccharides, or it is interacting with the phosphate groups on the DNA. So whether it's a DNA binding drug, whether it's a protein binding drug, whether it is lipid binding drug. So I can identify the molecular interactions also using this database. And when I'm talking about the interaction, we are strictly talking about the docking and the screening part. Now, what is docking? I told you in earlier lectures also. What is docking? I am docking a target. I have a target and there is a site on this. If I want to dock, like in harbor, when we are docking a ship, what it is happening? The hook is coming and attaching in the vacant site, and then the binding is happening. This is called as docking. Now we have different molecules. So a molecule which resembles in structure the site of that binding will be able to come and attach in that particular site, and this is called as binding site. And in docking, what we assess, in docking, what we examine. In docking, we examine what are those small ligands that can interact with this particular molecule. If this is a protein, that whether they can interact with this binding site of the molecule. Now, if I have 5 different molecules that are binding, how I will categorize? I will categorize based on the affinity of these molecules to bind, and that is called as computing the binding affinity. And then I optimize based on the position, whether by rotating, if the binding affinity is increasing, or by adding a certain group, suppose the complete binding affinity is increasing. So that is the part that we do in docking to optimize. By any optimization, can I reduce the binding energy? Can I reduce the delta G value so that a much stable binding can be achieved? Much stable binding can be achieved. That is the whole soul motto. That is the whole soul motto of optimization. So what we are doing, we are docking the small ligands to protein so that a ligand which is able to bind to a protein will inhibit that particular protein function. Now that binding can happen at catalytic site also, or it can happen at allosteric site also. So the small ligands which are binding to protein, that are of utmost importance to industries. Why? Because those are the molecules that are first picked up as the probable drug candidates, So apart from this, if this is the protein of interest, This is the protein which is required to inhibit. Then by docking, you may also identify that apart from this region, is there any other surface area which can be targeted? And if that can be targeted, then it again opens up a new picture of drug discovery. So that is how, that is the reason, that is the result what is achieved after docking, and that is called as virtual screening. That after docking, you have to identify that whether there are certain pockets available, whether the binding is, whether we are talking about the rigid binding or whether we are talking about the flexible binding, whether in rigid form, when we're talking about the rigid binding, whether the complete ligand is what we are talking about, or in flexible, whether that ligand is break down into pieces and then we are docking them individually. What happens is that sometimes what we do, the ligand which is to be docked here, first we are docking it completely. That is rigid docking. I opened it up. I first dock this particular part, then I dock this particular part. So what I have done, I made it a flexible docking, and that is what we do. So the structure wise, when we're talking about the structure part, then you must be aware of the structure database, that is PDB, protein data bank, which is used to take out the structure of protein molecules, which are deciphered based on your NMR or X-ray crystallography. So the PDB was the first structural database which was established in 1971. Then in 1998, 2 decades later, it was termed as RCSB, and till now it is running in that particular scenario because based on research collaboratory for structural bioinformatics. And then in 2013, almost 10,000 structures were deposited. Almost 10,000 structures were deposited, and till now, if we're talking about 2021, then that number has increased from, you say 10,000 to a lakh, 10,000 to lakh. It has reached to the lakh in 2014 itself. So continuously, we the scientific community is continuously putting up ahead the structures of different proteins, and it's because of the availability of that structural data that we can use in bioinformatics based studies or in silico drug designing to identify different targets. To identify different targets. Now, the main question what people ask me, why we need to go for experimental procedures? Why cannot we predict the accurate structure of the protein, how it will be folding in vivo condition? Can we predict the same in silico also? Then you should be able to know the basic problem. The basic problem. I can easily predict the secondary structure. And then from predicting to secondary to tertiary part, I need to incorporate many parameters. Many parameters. See, our cell is the best engineer, which is taking multiple parameters and folding the cell, and that folding is happening in fraction of seconds. In fraction of seconds, if I tell you that a protein folds, then a protein folds in 1/10 of the microsecond. If I'm talking about, that is on a million microsecond time scale. So to predict this short duration of work, it will take multiple memory and multiple CPU power to predict that particular thing, and that is the main problem. That is the main problem. That assignment of domains. That is the basic problem. That is the basic problem. So predicting a structure is one of the biggest. What you can see. I have a protein sequence. I will go for alignment. If it matches to a already known sequence and its structure is already identified, then I'll say that my sequence may also fold like that protein. By that, please don't take me wrong. I'm not saying that it is not possible. It is possible. Softwares are there to build the three-dimensional structure also. But then again, one should be able to understand the problem first of all, that what is the problem? Why folding is taken as the complex material when we are talking about in silico drug design, then it is classically defined as Leventhal paradox. It is classically defined as Leventhal paradox. What is Leventhal paradox? See, if I have 100 amino acids, they will be forming 99 peptide bonds. Now, if you know the Ramachandran plot, if you know the dihedral angles, then phi and psi, when we are talking about phi and psi, for 99 peptide bonds, we have 198 different phi and psi bond angles, right? And if we are talking about three stable conformations, let's talk about only three possible stable conformations, then these many bonds will be taking 3 raised to power 198 possible conformations. 3 raised to power 198 possible conformations. And we are talking about, let's say, a single conformation. A single confirmation is taking a definite time, that how much time will it take to predict all the confirmation and then pick one appropriate, right? So if I take this particular reaction at nanoscale also, suppose I am giving you nanosecond time to a single reaction, then to accommodate and to predict this much sample size, it will take very long time, very long time. And this is called as paradox, that how our cell is able to fold it correctly with this much of probability. And that is the thing which we have to mimic, or we have to mimic or simulate. And that is the difficult part. That is the difficult part. Now, see, proteins, why that paradox was given? It was given when we are saying 3 raised to power 198, we are not predicting any driving force or any directional force. We are going for random conformations. But when we are talking about protein folding, there are many forces that are coming into play: the disulfide bond, the hydrogen bonds, the hydrophobic interactions. So all those forces reduces the random conformation, of course. But then again, it is difficult to predict how those chaperones and the environment that is required for the folding, how are they working? How are they working? So naturally, when we're talking about general consensus from secondary to tertiary structure, from secondary to tertiary structure, the prediction is based on the understanding the working of chaperones and the post-transition processing, the environment change, the random approximation of the delta G values. So that is the reason. That is the reason when we are talking about the complexity and diversity, you have to understand that what is the potential of in silico and what is the reality actually. So an average protein size, if we say that it is of 300 amino acid, then if we are talking about 300 different amino acid, and we have 20 amino acids in total, so the total possible sequences that can be predicted is approximately 20 to raise power 300, right? It is more than this particular scenario. Why? Because we have humans as complex engineers, and we have more than 30,000 proteins, which are folding again and again, and if you are talking about 30,000 proteins, you can multiply this by 30,000, and that much prediction is practically is not achievable, right? So we have got to design and decipher things based on the potential and based on very clear cut understanding, taking into account the evolutionary part. What is convergent evolution? Convergent means we are converging to the same point, and divergent, we are diversifying from a common ancestor. So if you are able to identify the nearby ancestor, you may predict that how the fold will be happening. So the limited folds will be there when you're talking about the divergence part, and whether you are talking about a certain force that will be favoring the convergent evolution, that will be forming a common folding pattern. So why this folding is difficult to do so? And that is the thing that I told you, that if it takes a nanosecond to stimulate a single size protein, then if you're talking about the i7 processor, right? i7 processor, that will take around 30 CPU per year, 30 CPU years to simulate the complete folding pattern, and that is very difficult. So what is happening? What is the solution? The solution is homology modeling. See, if there is no structure for your protein, you need not to be let down. What you can do, you can go for the alignment part. You can search the sequence alignment. and eventually you will find the closest match, and you can predict that my protein can also fold like the closest protein, and that is called as homology modeling. You first search for the sequence alignment. If it's not there, you search for the secondary structure alignment. If it's not there, then you will try to overlap the three-dimensional structure with the already known structure, and you'll see that yes, this is the function, right? And that is called as homology searching or sequence alignment. That is the first step of protein structure prediction that I always go for homology modeling. Then we talk about the threading or ab initio modeling. So first is your homology model. So what is happening at, there are only two assumptions when we are talking about the protein structure prediction, that the structure of protein is solely determined by amino acid sequence only, and there is no external factor. And with evolution, the changes in the structure are very slow because evolution is slow process. So if I found a nearest match, then the distance between these two will be very slow, very less. Why? Because evolution is slow, and my unknown protein will fold as the previously known protein. And that is the, these two are the basic assumptions. These two are the basic assumptions that we're talking about. Now, just to give you an example. So, human protein, there's a human protein without a structure. When we went for a match, we observed that the sequence alignment is showing us very good results with a thermotoma maritima structure. That is a glycosidase. Let's talk about the glycoside. So it is matching the glycosidase. So what we did, we took this glycosidase as a template, took this glycosylate as a template and tried to put our protein sequence on that template, and we observed that our model is also folding similarly as the glycosidase model. The red is your unknown and the green is your glycosidase. So if you see the overlapping, because of the overlapping, it was able to accumulate one on the other, and you will see majority of the portions are folding in similar manner, although we have certain dissimilarities also. And that is how we predict. So what are the models? You can go for Swiss model also. You can start modeling. It's a freely available platform. You can go at the expressive Swiss model. You can start modeling the protein here. You can add up your protein sequence and you can go for structural prediction. Then we have one of the prominent database platforms, the web-based tool that we use is Fire2. Now it's the second version that we talk about. This Fire2, what you can do, you can incorporate your amino acid sequence here, and you can go for modeling, and it will give you different structures. And based on that structure, you can go for again go for modeling. Then we have genome 3D, that is again very much used if you are talking about the annotating genes with structure, the genome and the complete structure annotations you are talking about. Then you can use this particular structure. Now, genome 3D data platform, when we're talking about it, it provides you multiple informations including it takes information from cath database, it takes information from scope also, scope. Then it is in line and can take the input of fire also. Then it has threader also, so that you can perform multiple things from using this particular database. And in fact, in the example, if you go to the page with a human, then it will give you the complete information, the structural knowledge of that particular part, and the tutorial will begin. So using the models, when you're talking about using docking and screening, first we have seen that how structure is predicted. Now, when we are talking about docking part, how it is done, then for docking, what you need? First, you need a target. So first of all, you have to choose the target. Then you have to prepare the target. You have to remove the unnecessary things which you don't want to ligate and dock. Then that is called as refining. Then among the complete target, you have to identify which is the binding pocket that you will be targeting with the ligand. And then you will try to fit the molecule in that binding pocket. Once you try to fit in the binding pocket with the ligand, you will be getting the scores of the binding energy, scores of the total energy. And then you keep on repeating this particular process with different chemicals, and you will get a table of the binding energy. The least delta G values will be giving you the stable binding energy, stable binding, and those molecules which you can pick up for going ahead in your preclinical experiments. So that is the part. First, you have to identify the binding pocket. So identifying the binding pocket, what it means, Identifying means that you have to identify if this is your binding pocket in red, or you are talking about the surface part that is surrounding that particular pocket to be used as a target. To be used as a target. So what is there? This is a structure. Complete. This is a structure. So what we did, we refined everything in the structure. We identified that this can serve as binding pocket. Now, how can you be so sure that this is a binding pocket? See, the moment you have taken that structure, the target from any database, it will give you the binding history. And it will also tell you that yes, this is the pocket that is actually binding to this particular divalent ion, or this is the pocket which is binding to a certain domain. Then you can have a notion before going for refinement that this might be the pocket of my interest, and that is how we identify the particular pocket. Then you have to prepare the ligands of the chemical database what you are using. You have to isolate the complete structure, make the library, extract extract the ligands from that database, and then the extracted ligase will be going for the docking. Going for docking, you can add the hydrogen atoms. You can optimize the structure in different database. When they are ready to dock, then you use them as ligand to dock against those molecules. So that is how the docking is done. So there are three types that we always talk about: rigid, flexible, and possible. Rigid is nothing but when you have fixed the structure of ligand, and this structure will be going for docking. Flexible is you have allowed the flexibility in the molecule so that when it is going for docking, if it is not docking in that manner, it can change the orientation and give you the result in this particular manner. Of course, the data output will be very high. Then possible. Possible means the ligand structure is rigid, but it can move spatially. It can move spatially. It can search the pockets different angles. So that's how the docking is done. When we're talking about rigid docking, the structure is fixed, the orientation is fixed, and then it will be docked with the atoms of the binding site. So that is the thing that one should be available. Then there are other alternatives also when we're talking about flexible and possible part. The flexible part, when we are talking about the rings that we're talking, these rings are flexible, fine. But in possible, these rings are rigid, but the ligand can move in the spatial form. The ligand can move in the spatial form, and that is all. So just to give you an example of avidin and biotin, just to give you example of streptavidin and biotin that we regularly use, right? Biotin is actually the ligand, and avidin is the protein. So this is avidin. This is avidin, and this is the binding pocket that we identify that the biotin can enter from here and can attach in this particular binding protein, and that is how we decipher. That is how we decipher that how these things will be going about. So when we are talking about which drug or which compound is actually one should consider, which compound one should consider that's the best suited part, is the best suited thing that we are talking about. See, there is this very clear-cut notion about it. First of all, the molecular weight should be less than 500 Dalton. If you're talking about the safety profile of a drug, it will be very less toxic. Then the lipophilicity, affinity towards lipid, should be very low. We measure them in terms of log P values. The log P value should be less than 5. Then the number of hydrogen bond donors in that particular compound should be less than 5, and the number of hydrogen bond acceptors should be less than 10. These four are the parameters that we use and called as Pfizer rule in terms of understanding the safety and efficacy part. And this is the thing which actually tells you that out of the complete data, what portion you have to take. What portion you have to take. This is in brief summary about the prediction of structure of protein, talking about the docking part. Now, next phase which I incorporated in this particular lecture, just to give you a glimpse, is biologics. What are biologics? See, that's a basic question. What are biologics? Typically, what everybody thinks is biologics. Biologics are related to when we talk about antibody mediated therapies, or we are talking about the immune based therapies or components of vaccine products, but if we talk about the technical aspects, if we talk about the technical aspects. Then biologics. Biologics can be made up of anything. See, we are talking about any biological material. Okay. What can be those biological materials? It can be sugars, it can be proteins, it can be nucleic acid, it can be antibodies, it can be the portion of the vaccine, it can also be the combinations of different things, the living entities. It can be a viable microbe, it can be a viable compound isolated from a microbial origin. It can be isolated from a plant origin. It can, so these biologics are not determined to therapeutic only. They can form different things. They can be of vaccine. They can be blood and blood components as growth factors. They can be used in allergenics. We can be using them in gene therapy or recombinant therapeutic proteins. So all those materials collectively are called with a single term as biologics. So they can be gene-based biologics, they can be cellular biologics. So that is how we understand biologics. So it's not that we are only talking about antibodies or vaccines in biologics, but for our lecture's sake, we will be focusing right now only on antibodies only because just take an example how the simulation-based biologic prediction is happening here. How simulation-based biologic prediction is happening here. So again, the safety and efficacy of biologics is very important. When we are talking about the safety part, then it's very clear that you are putting it inside a human body. It should not evoke a very high immune response. So the safety is primarily focused on the potential of that particular drug to raise the immune response. That can range from very mild to severe also. Very mild to severe also. And then when we're talking about the efficiency part, when we are talking about the efficiency part, that means that particular compound, what is the half-life when we're talking about that particular protein, or whether it is able to generate any kind of anti-drug antibody response, or whether that particular molecule what you have used, our body has mounted a response and has made an antibody against our drug itself. That is what profile one should be able to take into consideration. So when we are talking about the antibody part, what we are talking about? We are talking about using the antibodies in therapeutic settings. And those antibodies, what I am talking about, are monoclonal antibodies that we regularly make in the lab, right, for immunotherapy. So traditionally, monoclonal antibodies are made using mice, right? If I talk about the mice, then of course phages and ribosomes that are used in this particular settings. So it's okay if you're preparing in mice. There's no harm. There is no problem. It's good for research and development sector. It's good for your studies point of view. But when you are talking about transition from mice product to humans, that's a complete extrapolation that you are talking about. Whether the acceptability of that particular antibody will be same in terms of complex human physiology, that is unanswerable until unless it is found out. So when we are talking about the developing of antibodies for therapeutic use, there are very few requirements in silico terms. So most of the work that is required is actually done on the molecular biology level. So what is happening in silico part? In silico part, what we do in silico, we take the information from that molecular biology aspect, like gene editing expression system, and we refine them. We modify their function in order to in order to consider safety and everything. So if we talk about immune response, let's see. This is very common slide. Everybody knows it, right? So I'm talking about the B cell activation. What is B cell activation? The T helper cells are actually responsible for activating the B cells, right? They will be having an antigen. Antigen will be there. So what is there? See what is happening. These B cells are antigen presenting cells, right? So a B cell gets activated in terms of humoral response. The B cells, when it encounters an antigen, it gets activated. It takes up the antigen, and after taking it up, it presents the antigen on the surface via MHC molecule MHC2. And this MHC2 molecule will be recognized by the T cell receptor and the other co-stimulatory receptors of CD4 and CD40L, CD40 binding. And these bindings will ensure that the T helper cell is now activated. Now, once the T helper cell is activated, then it will start secreting the cytokines that once it is shown in yellow. Once these cytokines are released, then these cytokines will help the B cells to get multiply and to for clonal propagation and to multiply in mature into plasma B cells. So until and unless the cytokines are released from these T cells, the plasma cells are not formed. And once the plasma cells are formed, then these antibodies are released into the body, into the blood. And then these antibodies will search for the antigen to form a lock. And then once they attach with the antigen, then the complement based system will be activated. So when we are talking about the antibody structure, this is the real time structure. This is the structure that we talk about. This is immunoglobulin superfamily domains. This this is variable region, this is constant region, and in this variable region, the two domains that is present, these are nothing but they contains three CDRs, CDR1, CDR2, CDR3 complementary determining regions, which are actually the paratopes, which are actually the antigen binding domains, right? So what we are talking about here, let's see what we are talking about in terms of comparison. This is our antibody, right? This is our antibody. I am talking about the comparing the size of different proteins. Okay, so this is our antibody. Let's talk about when our antibody wants to bind to a viral pathogen like rhinovirus, which is a common cold virus. See the size of the rhinovirus and see the size of your antibody. Or whether we are talking about the other proteins like cyclohexogenase or the ribosomes or the DNA polymerase. So based on understanding the structure, I can engineer these proteins or I can engineer the biologics. When we are talking about the antibody, then let's say what engineering domain I am talking about. See, the chimeric antibody. The chimeric antibody is 75% human, what I have written. What do you mean by 75% human? See, although it will retain the mice variable domains, we are mixing these two. The mouse and the human antibodies are mixed to produce the chimeric antibodies, which is having the epitopes that were generated in mice, but the basic module, the remaining constant region is of humans. So 75% of the region you have taken from humans, and the epitopes actually, which are referred or which are generated inside the mice against a certain specific pathogen, those epitopes you have taken of mice origin, and thereby you have reduced the chances of not accepting by the human body. Or next is humanized antibody. The same case where 95% is retained, where you have only retained the CDR regions, In earlier case, it was 75%, now it is 95%, and humanized antibodies are the most acceptable form of monoclonal chimeric antibodies that are used in therapeutic settings. So what you are doing, you are just taking these two regions and you are keeping the remaining portions as such, and you remove the unwanted post-translational modifications. You focus on a very specific fragment crystal constant region, that is FC region. You have to clearly understand that this particular region will be responsible for eliciting the complement pathway and binding to the phagocytes also. So when you are talking about the drug part, you want to add a certain drug on an antibody, then it's the CH4 domain which is taken into consideration, where the drug should be binded, right? This is the region where the post-translational modification is modified. This yellow color region, the VL and the of the heavy chain and is VL of the heavy chain and light chain, the variable region of the heavy chain and the light chain. Here, what you do, you remove all the unnecessary post-translation modifications that are not required. And in the CDR regions which are present in these variable and light chain, what you do, you you mitigate the complete immunogenicity, and then you talk about engineering the antibody, and that's how you engineer the antibody. So just to give you a glimpse, that when we are talking about immunoglobulin G engineering IgG, we have 4 different isotypes, right? 4 different isotypes. So basic understanding of these different isotypes has to be there. See, if I ask you the which isotype you want based on your half-life. So the Ig1 has the longest half-life in the blood, whereas the least is of Ig4. Least is of Ig4. Then got the structure part, the effector function part, and then the hinge length part, then the potential in order to be getting manipulated, or what other different allotypes which are present. All these scenarios have to be taken into consideration before going for engineering an antibody, so that you engineer a putative allotypic antibody or isotypic antibody in terms of synthesizing in silico. So that is what we should understand, whether it is half-life we are talking about, or it's binding to certain receptor we are talking about, that is able to bind to a certain receptor. In that case, whether what we have predicted will be exactly binding or not. So all these scenarios, whether we are talking about in the neonatal form or whether we are talking about the binding in terms of adult form, so the structural part, whether your antibody will be able to bind to the receptor, that you take into consideration. And that is how what we do. This is how what we do. We first take out the structure of the target, we synthesize the IgG, and we try to put this IgG and dock it with the receptor. If it docks perfectly, then our IgG, what we have synthesized, is working fine. If it is not docking properly, then perhaps we have to reconsider engineering it again. So that is how the function is going about. That is how different biologics are being synthesized, whether we are talking about in terms of T cell epitope or whether we are talking about in terms of B cell epitope. But the main aim is whatever biologic you are synthesizing through engineering or whether it is deimmunization, a simple thing is you are aiming to reduce the immunogenicity of that particular thing. Now, coming to the part of the platforms, to predict the T cell epitope, we have very good softwares. We have TC, the T cell epitope prediction software, where you can directly use the MHC binding molecule 1 or MHC binding molecule 2. You can see the IC50 values also. It will go on the sequence level engineering. Then you can identify whether what is the sequence of the T cell epitope, what is the amino acid sequence, and whether your one is weaker or non-epitopic region. So coming to the final conclusion, what softwares you should focus while working in this particular in silico modeling? For homology modeling, you can use this. Perhaps the fire is extensively used. For model views, PyMol or JMol, you can use anything. JMol is also wonderful software. For molecular simulation, Tinker is one of the finest. Then Chromax is there, Ember is there. For docking, freely available Autodock is there. Autodock Vina is also used by people. Then paid or purchase platforms are also there with multiple add-ons. Then graphical tools are there, or you can purchase the complete suits of the companies of Yasra, OpenEye, or Tripos. So that is the thing. So you have to understand which one is free and which one is costing you for a commercial use. So first of all, you have to understand the limitations and you have to understand that which function you are working on. So based on that only you can take the different parts and you can start working on that particular scenario. So that is the reason that we talk about that you should be very judicious in terms of choosing the right software and in terms of performing the right experiment. So for your further reading, I have given you these particular links, which are very well defined. In fact, the NPTEL is running the complete computer-aided design lecture. You can go through it, you can understand the concept, and then this review will help you perhaps understanding it much efficiently. So with this, thank you so much. Thank you for your patience listening. We'll be again meeting with another set of topic in the same lecture series. Till then, thank you and wish you a happy learning.